Hello, dear students. Welcome back to GDC. I hope my voice is audible to all of you. Please confirm. So, I am audible to all of you or not, and my screen is visible to you all or not. Hello, everyone. Welcome back to GDC. So, in this today's class, we are in this marathon series, we are going to discuss about the another 50 important MCQ, which is related to the subject pharmaceutical biotechnology and management. Okay, so after this, I will request to all of you join the batch of marathon series revision test batch and attend the test to evaluate yourself. Okay, so let's start. So the first question is micro propagation of the plant is carried out through. Okay, so what is first thing is what is micro propagation? Micro propagation is a technique where you can do certain manipulation inside the plant. So how you can do this micro propagation? Your option is cross fertilization, seed germination, uh, plant tissue culture, yeah, grafting. Yes, answer your question. So micropropagation of the plant is carried out through cross fertilization, seed germination, plant tissue culture, yeah, grafting. So I discussed all the things in the theory class. So what is micropropagation? So micropropagation is a technique in plant tissue culture technique where you basically manipulate certain things, certain genes inside the plant. So that is basically here called as micropropagation. So here the correct answer will be yes, option C. Very good. Option C is the correct answer. So plant tissue culture technique. So you can see micropropagation is a mass multiplication of desirable plant by culture technique is called as micropropagation. So after this, your manipulation, your plant will multiply in a very mass number. So that technique is called as micropropagation. Next question. The most effective method for producing virus-free plant. Very, very important question. I always tell you that this is very, very important question. So how, by which tissue culture technique you can produce a virus-free plant? Okay. So option is root culture, meristem culture, somatic embryogenesis, yeah, floriculture. Yes, answer this question. Hello, everyone. Welcome back. So answer this question number two. The most effective method for producing virus-free plant. So virus-free plant means, so in this method, if you produce a plant, that plant will never be affected by any viral disease. So for one question always asked in the exam, which is the first plant virus to be detected? So first plant virus is, please remember TMV. Okay, TMV stands for tobacco mosaic virus. Okay, you should also remember this. The first virus that is detected in plant is your TMV. So virus-free plant basically produced by which technique? Meristem culture. Okay, option B is the correct answer. So you meristem culture is basically is a undifferentiated cultivation of apical meristem is called as meristem culture, and this is the most effective method for producing a virus-free plant. It is the most effective method for producing a virus-free plant. In callus culture, you know the undifferentiated, unorganized mass of cell is callus, and this is basically a parenchymatous tissue with there are two hormones is required, oxygen and cytokine, and temperature should be of 22 to 28. Okay, already these things we have discussed. Next question. Formation of somatic embryos or embryogenesis tissue is directly from the egg plant without the formation of an intermediate callus phase is. So again, I'm repeating the question. Formation of somatic embryo or embryogenesis tissue. Okay, directly from the egg plant. Okay, without formation of an intermediate callus. Okay, so it is telling that from a egg plant, your direct somatic embryogenesis is occur without forming any intermediate. So that is called a somatic embryogenic response, callus response, direct somatic embryogenesis, yeah, premature germination. Yes, answer this question. So where it is telling that without formation any intermediate. So you will not see any intermediate, direct formation you will see. Yes, answer this question. Somatic embryogenesis response, callus formation, direct somatic embryogenesis, yeah, premature germination. See, So callus formation means what? So when there is a synthesis of any undifferentiated unorganized mass from an X plant, that is called as callus. But in case of callus culture, you will find different intermediate step. But here question is asking about without any formation. So when the question is asking you do not need any intermediate, then that is called as direct somatic embryogenesis. So please remember we call it as a DSC. So direct somatic embryogenesis is basically formation of your somatic somatic embryo without forming any intermediate. Without forming any intermediate. Option C is the correct answer. Somatic embryogenesis is the process, the regeneration of embryo from a somatic cells or tissue organ and regenerated as a somatic embryogenesis. Okay. If you see, there are two types, what the direct somatic genesis and indirect somatic embryogenesis. In direct somatic embryogenesis process, the embryos form directly from the cell of a explant without going through the unorganized callus phase. Whereas the indirect phase, embryogenesis involves there is an intermediate stage called as callus formation stage, which embryos are developed. Okay. Next question. The artificial or synthetic seeds are prepared by which process? Somatic embryogenesis and culture, somatic embryogenesis and immobilization, microprojectile and transformation, yeah, artificial pollination embryogenesis. Yes, artificial or synthetic seed, how you can prepare? Answer this question. Artificial or synthetic seeds are prepared by the following process: somatic embryogenesis, somatic hybridization and culture, somatic embryogenesis and immobilization, microprojectile and transformation, yeah, artificial pollination and embryogenesis. The artificial seed, you remember, artificial seed is basically produced through, yes, option B, very good. So those who are giving option B, correct answer. Somatic embryogenesis and immobilization. Let's see. So the somatic embryogenesis means what? Somatic embryogenesis is nothing but this is a process regeneration of embryos from the somatic cells or tissue or organ is regarded as a somatic embryogenesis. So artificial seed can be made encapsulated on somatic embryos. The embryos coated with sodium alginate and nutrient solution are deep inside a calcium chloride solution, and that calcium ions is rapidly cross-link of sodium alginate to produce a small gel beads, okay, which containing encapsulated embryos. Okay, then artificial seed like encapsulated embryo can be formed. Okay, so we have already discussed this. So somatic embryogenesis and immobilization technique basically used to synthesize artificial seeds. Okay, next, the technique of using very small metal particle coated with a desirable DNA in the gene transfer technique. Yes. So I told you, so small metal particle, and the in over the small metal particle, you have to do the coating of your desired DNA, and you have to enter the insert this. Okay, so this technique is called as what? Micro injection, biolistic, liposome mediated, yeah, micro electroporation. Yes, so liposome mediated means you will need a liposome. So inside the liposome, the gene has to, the desired DNA will be incorporated. If we talk about micro injection, here you have to do this direct injection, direct injection of desired DNA. Okay. If we talk about bioelastic, so bioelastic is the correct answer because in bioelastic, over a metal particle, you have to coat the desirable piece of DNA and that metal particle will be inserted. And electroporation, this is the electric mediated gene transfer where you have to change the voltage and that will leads to transfer. Okay. Next question. Electroporation or microprojectile or particle bombardment or bioelastic, this is a technique which using a very small metal particle are coated with a desired DNA in the gene transfer technique is called as bioelastic technique. Okay, DNA is bombarded into intact cell using a bioelastic device such as gene gun. Okay, next question. So you can join. So whatever things we are discussing here, you can do this. You can analyze yourself by attending the test. So for the attending the test, you need to join the batch. So the batch person you have to apply the batch code MAHA 26 MAHA 26. So they after joining to this batch, you will able to join this test and you can attend. So there is a MAHA revision test number 5 from test 28 to 35 on 31st January and at 9 p.m. So all are requested to join and analyze yourself by giving this uh, test. So I want to so all those who are joined this batch, kindly put a thumbs up in the comment section. I will check how many of you have joined the batch and giving the test regularly. Okay, chalo. Next question. Match the list one with list two. So here you have product and here you have which strains are used. Okay, you have to match. Okay, so first thing is dextran, bacterial amylase, glutamic acid, and vitamin B12. So for dextran, the species is, yes, Leuconstactus mesenteroid. For vitamin B12, the species is Streptomyces olivaceus. Okay. Next is for glutamic acid, the species is Brevibacterium species. Okay. And from dextran, it is uh, for dextran, it is for bacterial amylase, it is Bacillus subtilis. So Clostridium tetani is not because it is a pathogenic bacteria. Okay, it causes your tetanus. Okay, so this your answer will be one S. Sorry, one R option D. Option D is the correct answer. Those who are giving option D are correct. Okay, next question. So you can see for dextran, the species is Leuconstock mesenteroid, and Acetobacter species and Streptococcus mutans. For bacterial amylase, it is Aspergillus oryzae, Aspergillus niger, Bacillus subtilis, and Bacillus leachiniformis. For glutamic acid, Mycobacterium species, Brevibacterium species, and Arthobacterium species. For vitamin B12, Bacillus mesenterius, Streptomyces olivaceus, Butyribacterium letrigi, Micromonospora species, and Propionibacterium predilecti. Okay, next question. Match the following enzyme of column 1 with the respective function in the column 2. So this is associated with this question is associated with the biotechnology, recombinant DNA technology. So you have enzyme and you have to match with the column, second column. Okay. So DNA ligase, alkaline phosphatase, reverse transcriptase, and polynucleotide kinase. Okay. So alkaline phosphatase. So alkaline phosphatase, what is phosphatase work is removal of phosphate group. That is 2R. Okay, removal of phosphate group. Next is DNA ligase. So DNA ligase, I told you this is just act like a glue. We call it as a molecular glue. So what is its function? It will act by forming for at by at major function is to join the fragment DNA. So how it will join? So it will join by forming a bond between 3' and 5' phosphate group. So DNA ligase is work by forming a bond between 3' to 5'. So reverse transcriptase and polynucleotide kinase. Uh, okay. So reverse transcriptase work is to synthesize the DNA copy of RNA. So in case of certain virus, what happens? So the genetic material is RNA. So they basically synthesize complementary DNA, or we call it viral DNA from that RNA. So for that, reverse transcriptase enzyme is required because this process is carried out through reverse transcription. So reverse transcriptase is here required for the synthesis of DNA copy of your RNA. And last is polynucleotide kinase, which basically add the phosphate 5' OH at time. So one Q 2 R. Option C is the correct answer. Okay. Next question. You can see alkaline phosphatase, it removes the terminal phosphate group. DNA ligase forms a bond between 3' to 5' phosphate. DNA polymerase forms a nucleotide to form complementary DNA strand. Exonuclease 3 to remove the 3' end DNA. Gamma exonuclease for the remove the nucleotide from 5' end DNA. Polynucleotide kinase transfer the phosphate ATP from 5' OH end to the DNA or RNA. Restriction in the nucleus cut the DNA strand with specific recognition site. Reverse transcriptase synthesize DNA from the RNA. Tag polymerase use PCR and terminal transferase at nucleotide at the 3' end of DNA. Okay. Next question. Which of the following enzyme is necessary to make a accurate copies of vector DNA? Your option is RNA polymerase, DNA polymerase, DNA ligase, or reverse transcriptase. Which of the following enzyme is necessary to make a accurate copies of vector DNA? Okay, answer this question. So I'm saying very less number of students are giving answer. So I want every student to participate. So this is the reason why we are conducting this. Okay. So the correct answer is reverse transcriptase. Okay. So how let's see. So the reverse transcriptase, your enzyme, what it do, the first step is of synthesis of DNA copy of RNA using an enzyme called reverse transcriptase. Okay. So DNA product here called as cDNA because this is called as complementary DNA, which is synthesized which in response to the RNA. Okay, the enzyme RNA-dependent DNA polymerase or simple reverse transcriptase is responsible for the formation of this state. Next question. In DNA replication, the newly added nucleotide is joined to the growing DNA strand by enzyme. DNA polymerase, DNA ligase, restriction endonuclease, or reverse transcriptase. Answer this question. DNA replication, the newly added nucleotide strand is joined by growing DNA strand by which enzyme? Yes. So first thing is DNA ligase is joining a fragment DNA. So that's why it is the option is not. Restriction endonuclease is for the cutting of the DNA. Reverse transcriptase synthesis of complementary DNA from RNA. This is also not the answer. The correct answer will be DNA polymerase. So DNA polymerase will help for the replication of the addition of the nucleotide to the newly added strand. Next question. Polynucleotide kinase transfer ATP from 5' OH end and DNA and RNA. Reverse transcription synthesis of DNA and RNA. Already with these things we have discussed. Next question. Which of the following test best technique for detecting HIV, human immunodeficiency virus? Real-time PCR, Whittle test, reverse transcriptase PCR, or polynucleotide polymerase chain reaction? Answer this question. Very, very simple question, but little bit twisted. Yes. So what is uh, genetic material in HIV? So genetic material in HIV is your mRNA. Okay. So as this is the RNA present in as a genetic material, so you need to do this reverse transcriptase to synthesize your cDNA. So then which technique will you do? You will use reverse transcriptase PCR, na? So option C is the correct answer. Next question. Making multiple copies of desired DNA template is called as. Making multiple copies of desired DNA template is called as cloning, transferring, recombinant DNA technology, or genetic engineering. Yes, you are forming multiple copies of the DNA. So this is called as very good. Option A is the correct answer, cloning. So cloning means just like a, so you do a photostat or Xerox copies you do. So you place a paper and you type it 20 copies. So then automatically 20 print will become. So it just this process is just work like that. So this cloning technique, you produce multiple copies of the same DNA. Okay. Next, making multiple copy of desired DNA template which is genetically identical. Okay. Next, this can be natural or artificial. Next question. What is plasmid? So plasmid is a macromolecule involved in the protein synthesis, circular piece of duplex DNA, a hybrid DNA that is formed by joining piece of DNA, yeah, endogenous secreted by one type of cell, endogenous substance secreted by one type of cell. Yes, answer this question. What is plasmid? So plasmid is basically a extra chromosomal DNA of any type of uh, prokaryotic organism, extra chromosomal, okay, extra chromosomal, double stranded, circular DNA, okay, extra chromosomal, double stranded circular DNA, which is present in case of bacteria. And what is the major role? So, plasmid is majorly used as a vector, okay, it is majorly used as vector. So it is most commonly used bacterial cloning vector. Its size is around 1 to 500 kilobase pair. Next question. The plasmid conferring drug resistance are called as. The plasmid conferring drug resistance is called as R plasmid, col plasmid, crib plasmid, yeah, degradative plasmid. Drug resistance. So R for resistance, R plasmid. So R plasmid stands for the resistance. So drug resistance plasmid. Okay. So option A will be the correct answer. Okay, let's see what is this R plasmid means. So plasmids are basically small circular double-stranded DNA. Some plasmids provides the genes that provides a resistance to different antibiotic and other drugs. So this type of plasmid is called as R plasmid or a resistance plasmids. Okay, next question. In genetic engineering, what is the function of origin of replication? To ensure plasmid stability, to allow plasmid to replicate in the host, to provide a selectable marker, yet to integrate the plasmid into host genome. What is the role of ORI? So ORI stands for origin of replication. So origin of replication is the site where the replication process is basically started. So okay, so this basically allows the plasmid to replicate inside the host. Okay, it will help the plasmid to replicate inside that host. Okay, so this is just like a marker. So when this ORI found, so from that process, from that place, your origin will be your replication will be started. That's why we call it as the origin of replication. Next question. So, what happens in case of certain bacteriophage when their desired DNA is incorporated into the bacterial chromosome? The bacteriophage is immediately lyses the bacterial cell. The bacteriophage DNA remains permanently in the chromosome. The bacteriophage stop replicating. The bacterial chromosome get degraded. Yes. What so so the process of transferring the gene to the bacteriophage is called as transduction. All should know. Okay, so transduction process. So in transduction process, when the bacteriophage carrying the desired piece of DNA enter into the bacteria, what it will do? So it will remains permanently in the chromosome. Okay, that piece will be automatically incorporated into the host genome and it will start producing protein. So for that reason, it should be remaining permanently in that host. Okay, option B is the correct answer. Okay, so bacteriophage is a simple bacteriophage that replicates within the bacteria. In case of certain phage, the DNA get incorporated in the bacterial chromosome and remains there permanent. Okay, it remains there permanent. Next question. So you can, our new batch for to So, yeah, what is the ideal time to prepare for your GPAT exam? The ideal time to start your preparation is today. Okay. So, as you know, GDC is always taking care of you from the very beginning to taking this in consideration. We have launched the prime batch for GPAT 2027 and 28. Okay. So, where the value of prime batch, how your seniors, how your friends, how your classmates have succeed by joining this GDC prime batch. You can ask them, and there is a lots of extraordinary result we have produced in here. Okay, so you can join this batch GDC for those who are preparing for GPAT 2027 and 28. This is the right time to start your preparation. Okay, so for joining, you can download the GDC class app from the store, and where you can uh, joining. Okay, next, select the correct sequence of nucleic acid blotting. Okay, so nucleic acid blotting is basically for the detection of nucleic acid. So what is the step? Stranging wash, hybridization, pre-hybridization detection, hybridization, stranging wash and detection, hybridization, detection, stranging wash, pre-hybridization, stranging wash, detection, pre-hybridization, and hybridization. Okay, so let me explain you. So this is a technique for the detection of either DNA or RNA. Okay, for detection of a DNA or RNA molecule. Okay, so first step is, so whenever you start doing this blotting technique, so you need to transfer the DNA or RNA into the membrane. So once you transfer it to the membrane, so first step you do is pre-hybridization. Okay, pre-hybridization. Okay, that means so you need to treat to the membrane with the probes. Then after pre-hybridization or blocking step, we call it blocking, unnecessary blocking. Then we do hybridization. Hybridization means you have to add this complementary probe. Okay, hybridization. So after hybridization, you have to do this washing to remove unnecessary binding. After washing, you have to do this detection. So how you will do detection? You have to do detection by adding the complementary probes. Okay, so then you will going to detect that species. The first thing is pre-hybridization, then hybridization, then washing, then detection. Okay, so option B is correct here. Next question. Southern blot is used. So I told you, so we have three type of blotting. One is southern blot, one is northern blot, one is your western blot. Okay, for southern blotting is used for the DNA, northern blotting is for the RNA, western blot is for protein. So southern blot is technique. This is for the hybridization of DNA molecule. Okay, next question. For protein detection, most commonly used probe is. So for protein, what we use? Interferon, antibody, lectin, या antigen. For Western blot, I told you, so Western blot is a technique. This is for the detection of quantitative detection of protein in the biological sample. So Western blot basically use the probe as primary antibody. Use probe as primary antibody. Okay. Next question. Virus-mediated transfer of host DNA from one cell to another is known as. So virus-mediated transfer of host DNA from one cell to another is called as transcription, integration, transduction, or transformation. Yes, answer this question. Virus-mediated transfer. So let me simplify this question for you. So here you are using bacteriophage. So when you using a bacteriophage, so this technique is called as very good offensive transduction. I told you na. So the process of transferring gene by help of a bacteriophage or any bacteriophage is what? It is a virus. Okay, virus. So that is called as transduction. Next question. Which of the following test tube method is used for amplifying selected DNA sequence? Okay, you are using for amplification of selected DNA sequence. Okay, your options are polymerase chain reaction, redox potential reaction, oxidative reaction, yeah, reduction reaction. Yes, very easy question. So you are using for which purposes? You are using for amplification of what? It's selected DNA probe. So which technique is called an amplification technique? Very good. PCR technique. So I told you na PCR is called as a cell-free amplification technique. It is called cell-free amplification technique. So that's why we call it as a test tube. Here you are not required any cell for the amplification process. Next question. Which of the following step in PCR involves DNA polymerase synthesis, a new DNA strand complementary to the template? Denaturation, annealing, extension, or termination. So we are telling that new DNA strand or complementary strand synthesis. So denaturation, there is a breakdown. So no. So what is annealing? So annealing means joining of the primer. So that is also not. Termination, there is no termination such step. So extension is there. So extension is step where the newly synthesized DNA strands can be synthesized. Okay. Next question. During the denaturation cycle, the polymerase chain reaction temperature rise to. So I told you, so during denaturation or breakage, you need to rise the temperature to maximum. So what should be the temperature range? It should be 90 to 98 degrees centigrade. Okay, so first step in denaturation, you need more energy or more temperature for the breaking. So that's why this your temperature needed will be around 90 to 98 degrees centigrade. Next question. The amplification by the polymerase chain reaction uses Thermus aquaticus DNA polymerase, DNA topoisomerase, RNA polymerase, yeah, RNA helicase. Answer this question. So for the amplification process, so which uh, uh, polymerase enzyme you are basically using? Answer this question. DNA amplification by polymerase chain reaction uses Thermus aquaticus. Okay, so I told you the traditional fragment that is used for the PCR is called as Leon fragment. Okay, Leon fragment. The major problem associated with this Leon or traditional polymerase enzyme is it is not temperature stable. Okay, they are not thermostable. That's why after each completion of the cycle, you need to add this enzyme. So to overcome this process, Thermus aquaticus DNA polymerase has come to the picture, which basically we can use for multiple time without adding it within the cycle. Okay, next question. Hybridoma technology is widely used for the callus culture, organ culture, monoclonal antibody, and attenuated microorganisms. Why hybridoma technology is used? I told you hybridoma technology is used for the production of monoclonal antibodies. Very good. Option C is the correct answer. Okay. So, monoclonal antibodies are basically produced by using this hybridoma technology. Next question. Arrange the following step in a sequence of the order for producing recombinant insulin. During the formation of recombinant insulin, which step you will perform? First, fusion of A and B chain for disulfide, cyanogen bromide treatment to remove methionine and beta galactosidase, introduction of A and B chain into a plasmid containing beta galactosidase gene, and synthesis of A and AB chain in the uh, E. coli. So first step is this. So you need to introduce the A and B chain gene to the plasmid to bacteria. After you entering this, now it will start synthesizing the AB chain. So when this AB cell is synthesized, you need to apply with the cyanogen bromide treatment, okay, to remove the methionine and beta galactosidase. And last step is used for fusion. That means your RSQP. Okay, RS RSQP. Option C is the correct answer. Next question. So 50 days model paper for every day for practice GPAT 2026. You can apply it from the timing between 2 p.m. to 9 a.m. So 125 MCQ. So to join this, you need to join the batch GDC 26. Okay, batch code is GDC 26. Okay, you need to join. So go and join this batch and start practicing your model paper from now. Okay, so this is very, very important for your to these mock tests are very, very important for your upcoming GPAT exam. This will give you a boost or confidence to prepare for the exam. Okay, next question. Which of the following statement is false about interferons? So interferons are the cellular glycoprotein produced by virus-infected cells. We need to find out the false statement. Interferon have no extracellular or no extra virus activity. Interferons are the virus specific agent that can interfere with the DNA and RNA virus. They are produced as a potent broad spectrum antiviral. So you need to find out the false statement. Okay. So which of the following statement is false? So here the correct answer is option C. So why? Because interferon are specific agent, they can act against both DNA and RNA virus, not all. It should be and. Okay. Next question. Gene therapy refers to the process of gene therapy. Gene means your normal gene. Therapy means you are using gene for a therapeutic purpose or any for disease condition. So this process refer to us identifying a disease-causing genes and activating them for therapeutic benefit, increasing the expression level of the set of genes which involved in given disease in affected cell to the selected modulating agent, transfer a new genetic material to the cell of an individual therapeutic benefit, remove the protein corresponding to the disease-causing genes from the cell of an infected individual. So first thing is gene therapy means what? So gene therapy means this is a techniques. What happens here? So here you are inserting a gene into a host for a rectifying a disease state. Okay. So that's why the name therapy is come. Therapy means treatment. So you are using gene for a treatment in different disease. So here the correct answer will be option number C. Because here you are transferring the genetic material to the cell of an individual for the therapeutic benefit. Okay, which benefit? Therapeutic benefit. Next question. The largest gene of human. This question I asked in GPAT. Largest gene. So largest gene is dystrophin, titin, insulin, yeah, phosphofructokinase. Yes, answer this question. The largest gene in human is dystrophin. The largest gene in human is your dystrophin. Option A is the correct answer. So dystrophin is the largest known human gene of 2.4 million bases. It is the major gene associated with your muscular dystrophy disease. This causes basically mutation of the gene coding for the protein dystrophin. Okay. The gene with much smaller size is insulin. Titin is the largest known protein. Okay. This is the largest known protein. Next question. One of the following is confirmed by DNA diagnosis stage. So, in which of the disease you can detect by DNA diagnosis stage? Hyperuricemia, cystic fibrosis, acute pancreatitis, या hyperlipidemia. So you can detect by uh, DNA diagnosis. So DNA diagnosis stage, you basically detect which disease, that is your cystic fibrosis. Okay. Cystic fibrosis is a disease where you can diagnose the disease on the basis of DNA. So there are other diseases also, like your Huntington's disease, Alzheimer's disease, type 2 diabetes, Parkinson disease, obesity, certain cancers, fragile X syndrome, Gaucher muscular dystrophy. These all things can be detected by DNA diagnosis method. Next question. Which of the following statements are true about prions? Prions are the type of infectious protein. Prions contain single-stranded RNA. Creutzfeldt-Jakob disease is not caused by prions. Prions are even more difficult to destroy than the bacterial spores. Answer this question. You have to find out which is the uh, correct, which statement are true. Here you have to find the correct statement. Prions are a type of infectious protein. Yes, it is correct. Prions contain single-stranded RNA. No, it contains DNA. Creutzfeldt-Jakob disease is not caused by disease. No, because Creutzfeldt-Jakob disease is caused by prions. Okay, prions are even more difficult to try. Yes. So option A and D are correct. Yes. Option C is the correct answer. Next question. So the anchor batch for GPAT 2026 is currently going on. Those who want to rapid revision in short period of time, they can join. Okay. So where you can get your things revision with a systematic plan and within a stipulated time period. You can do your all the revision for your GPAT exam. Okay. Chalo. Next question. How customer bias about the product which will influence the marketing communication? Positive effect, negative effect, no effect, or both effect. How customer can bias based upon the about the product. So so customer can be biased due to both your positive and negative effect. Option D is the correct. So because if you will see any positive effect on that product, or suppose this product is, let's suppose I take an example of a beauty product. So I am using a beauty product. I will recommend my friend that this product is very useful. You can use. So then that person is started using, and that person will come tell to the next person. By this also increase. Negative effect seems similar. If I'm using that particular product, I will recommend that. So this is not a good product for your skin, so you don't. So similarly, this message also transfers. So that's why both positive effect and negative effect can be matter. Next question. When a communication takes place among the member of some group, it is described as central communication, lateral communication, upward communication, or open communication. Yes, communication takes place among the group. So if there is inside the group member, if the communication is going on, that is called as lateral communication. Okay. So downward communication means follow from the managers to subordinate. Okay, that is called as downward. Upward communication means follow from subordinate to manager. Horizontal or lateral communication means exchange of information, ideas, or message among the individual or department who are the same hierarchy level within the organization. Next is external communication, involves communication within the people outside the organization. Next question. In a new product development process, after analysis of business, next step to be taken is test marketing, penetration marketing, brand marketing, and individual marketing. So once the product is developed, so next step what you will do. So before doing any product marketing and all, you need to do first the test marketing. You have to check what is the status of this what will be the status of the product when it will launch to the market. So you first analyze the market value and everything. So that's why the correct answer is test marketing. Okay. Next question. Shift from try my product to prefer my brand marketing strategy is done at what stage of product life cycle? Introduction, growth, maturity, या decline. So try my product to prefer my brand. Okay, so when I am telling prefer my brand means I have list of products. So try my product I'm telling means I have a single product. So that is the difference. Try my product means I am referring about a single product. Try my brand means I'm telling about the list of product inside my brand. So that is basically we uh, it's recommended when our product or when our brand is on a high growth state. Okay. Next question. Product and dash and promotion force 4 P's of marketing: product, price, place, product, place, and promotion, product, process, and promotion, production, process, price, production. So you need to know this 4 P's of marketing. The 4 P's of marketing. First P is product. Second P is your price. What price you are going to sell. Next is what place. That means what you are targeting, the targeting group of location or group of people. Or last is promotion. How you are going to promote your product. So these are the 4 P's of the your product. Next question. You can see product, price, place, and promotion. Next question. Which of the following represent a complete list of products that are offered by a company for sale? Product variety, product mix, product item, या product line. Which of the following represent the list of products that offered by a company for sale? Okay. So you're telling that from this you have to find out which is the where you will get all the list of products. For suppose I am a supplier of particular product. So when I will give a list, so that will consisting of all the product that is there in my in our brand and with their list price and discount and all. So that is called as product mix. Okay, product mix contains all the information about the product of a particular brand. Okay, so product mix is concerned with combined combination of the product offered by free for a sale. Okay. Next question. The process of establishing a product in the mind of targeted customer is called as product positioning, product differentiation, product targeting, या product segmentation. So that means you are telling that you are repeatedly entering this product marketing brand name or these are things inside the customer mind by means of different ways, like your advertisement by mouth to word to mouth promotion. So if you are doing this, so that is called as what? That is called as product positioning. You are basically positioning that product in the customer's mind. Okay, so option A will be the correct answer. Next question. So product positioning is the process of creating an image of the product in the mind of a consumer. So this question have asked 2 times, GPAT 2022, GPAT 2021. That I am basically occupying a distinctive position in the mind of a customer. Next question. Which of the following is not one of the function of management? Planning, controlling, organizing, or centralization. Which is not planning? Yes, you need to plan. Controlling, yes, you need to control that particular product or anything. Organizing. So obviously, you need to organizing that particular product when you enter into the market. Okay, but centralization is not a part of your management function. Okay, centralization is not a part of your management function. So planning, directing, organizing, controlling, satisfying, and coordinating. Okay, these are the function of the management. Very, very important chart. You can remember. Question can be asked in your next upcoming GPAT exam. Next question. A reporting relationship in which an employee receive a order from and report to only one supervisor is known as unit of command, centralization, decentralization, a line of authority. So when you are receiving a order, you need to report only one supervisor. So that is called as unit of command, centralization, decentralization, a line of authority. Yes, answer this question. So this is called as unit of command. So how that means, for suppose you are an employee you have only single manager. Okay. So you have to report this single whenever the product will come and you need to remember. For suppose you have 10 managers, but you are working under a team. Okay, for suppose you are working in a X team. So in X team you are working. So the manager who is the manager who is manager of X team, you need to report him. Okay, that is called as unit of command. Okay. So unit of command means employee should receive order from only one manager. So you have to report everything to a single manager, not a multiple person. Okay. So that is called as unit of command. Next question. Henry Foyle's principle spirit the crops means what? Corporate objective, group objective, team activity, yeah, team spirit. Foyle's principle. Spirit the crops. Yes. The meaning of spirit these the crops means, so you can guess from the word spirit, that is for the team spirit. A spirit the crop. So it basically highlighting about the team spirit, how you are motivating your team. Okay. So management should be strive to create unity, morale, and cooperation among the employees. So this is our Navigator series, which you will get your all the essential books or modules for your GPAT examination prepare. 15 GPAT modules, 6 MCQ banks, and one previous year. Okay. Next question. Intellectual property transfer is essential part of plant maintenance, technology transfer, quality control testing, yeah, warehouse. Intellectual property transfer. For suppose simple thing, I'm giving an example. So if for suppose you have a good command on your organic chemistry, okay? So you for suppose your friend asks that he please explain me with this topic, this name reaction, okay? So you explain this. So what you transfer here? You transfer here intellectual property. So this basically called as technology transfer. Similarly, companies or manufacturing companies they do like that. So if they have particular technique, so they will transfer this technique another company on the agreement of this intellectual property. Okay, so that is called as technology transfer. Okay, so technology transfer it is called. Next question. In which of the method an order of a fixed number of item is placed every time an inventory level falls to a predetermined point? This is called an ABC method, maximum to minimum method, open to buy method, yeah, economic order quantity. Yes, answer this question. In which method, so for suppose you have 10 items, for suppose 10 drugs, so when that list of number 10 drug is reduces, you need to order. So this process is called as what? ABC method, maximum to minimum method, open to buy method, yeah, economic order method. So this question was asked in GPAT 2014. Simple. This is called as EOQ. That means economic order quantity. So in economic order quantity means when the product level comes to a certain level where predetermined point, that means up to when this level is come, you need to immediately order that product. So that is called as economic order quantity. Option D is the correct answer. So economic order quantity is a mathematical tool for determining the order quantity that minimizes the cost of ordering holding inventory. This is based on EOQ. ABC method analysis is basically classifies the items or activities according to their relative importance. And BED method analysis is based on the shortage of the values and shortage cost of the item. BED stands for vital, essential, and desirable category. BED analysis. Next question. An inventory turnover of a year is considered to a satisfactory 4 to 6 times, 6 to 8 times, 1 to 2 times, and none of the above. In a year, how many times your inventory? turnover should be carried out to consider it as a satisfactory? Yes, answer this question. Because this type of question will ask in your exam. These basic fundamental questions. That means that they will check your analyzing capacity, how you can analyze the things. So an inventory turnover is basically if that means how many time if you'll order a more product or you will get a order from particular product in a year this much time bulk order, then you consider it as a satisfactory. That means around 4 to 6 times. If it will occur 4 to 6 times, you will consider it as a satisfactory. Let's come to this. So inventory turnover is a measure of number of times inventory is sold or used in the time period in a such a year. It is a ratio of showing how many companies inventory is sold and replaced order over a period of time. If it is 4 to 6 times, it is called a satisfactory. Although result vary by industry, typically manufacturing company, it is 6 to 8 inventory turnover per year. Okay. Next question. The good management principle revolves around the 3R. So what are the 3R of good management principle? Yes. Answer this question. For suppose you are a HR, okay. So what is your good management principle? So you need to think very carefully about three things. First thing is R. R stands for reward. So if a employee is doing performing well, you need to reward that employee. So next R is recognition. So if that employee is extraordinary, you have to recognize that. Okay. And as next R stands for responsibility. Responsibility. You have to give the responsibility or distribute the responsibility equally among your employees because this will not create any type of chaos within the company. For example, you are giving lot of work to a single employee and not giving any opportunity to other employees. That will create a bad management. Okay. So these things you need to keep in mind. Option C is the correct answer. Reward, recognition, and responsibility. Next question. All the following aims of sale promotion except inducing customer to purchase more item, to attract new customer, to increase the taxes on the product, to popularize the brand name. Okay, except. So all are correct except. So you will induce the customer to purchasing your product. Yes, that is absolutely correct. You will going to attract the new customer by giving any offers and all. That is absolutely correct. You have to popularize the brand name. That is also correct. But if you increase the taxes on your product, then this will reduce a financial burden to the customer. Na? That will reduces the purchasing frequency of purchase. So that's why option C is the incorrect answer. Next question. So how is the objective to introduce a new product, to attract the new customer, to maintain old customer, to improve the public image of the field, to popularize the brand name with the product, to assist the salesman and the dealer to induce a customer to purchase more items. Next question. Which of the following is not a method of human resource development? Okay, which is not a method of human resource development? Career planning, job rotation, performance appraisal, yeah, industry strength. Not a method of human resource development. Career planning, job rotation, performance appraisal, yeah, industrial strike. Obviously, you will do career planning for the better development, job rotation, performance appraisal. But industrial strike is not a comes under your human resource development. This is not a good skill. Okay. Next, you can see what are the methods for human resource performing up, performance appraisal, potential appraisal, feedback, counseling, training, role analysis, quality of work life, organization development, reward system, job rotation, and career planning. Next question. Which of the following test is distribution free, does not require any assumption to made about the population following a normal or any other distribution? So this is which type of distribution is? Yes. This following test is a distribution tree. So distribution tree test is your Kruskal-Wallis test. So let's see what are these. So Kruskal-Wallis test is a non-parametric distribution-free test, meaning does not require any assumption that the population data follows a normal distribution. It is used to compare three or more independent groups of a single continuous ordinal outcome when the data violates the assumption required. So Fisher LST test and ANOVA are the parametric tests that assume normal distribution and homogeneity values. Student t-test, it is also a parametric test used to compare means of two groups under the assumption that is normally distributed. Okay. Next, which statistical test is appropriate for the comparing the means of two independent groups? Here is another independent. Here is another one. ANOVA test, pair t-test, pooled t-test, या sample t-test. Independent group. Let's suppose I am giving an example. So list of boys, average speed of aeroplane. So these two are independent value because here you are having a list of boys. There is no link. So if you want to compare, so statistical test, then you have to do this pool test. You have to pool the data. Okay. Next question. A hypothesis that uh, stipulating that there is no difference between the situation, group, and outcome. Okay, is called as null hypothesis, hypothesis of association, hypothesis of difference, yeah, alternative hypothesis. There is no difference between the situation, groups, and outcome. Yes, if there is no difference between the situation and outcome, that is called as null hypothesis. Okay. Null hypothesis, there is no difference between the parameter and specific value. Last question. Which of the following is used to test the interaction effect in a fractional experiment? ANOVA, chi-square test, correlation coefficient, or regression analysis. Test the interaction effect in a factorial experiment. So how, if you want to know the interaction effect, that means interaction between the groups, interaction between the parameters. So for that, which technique you will use? Yes, answer this question. ANOVA, chi-square test, correlation effect, yeah, regression analysis. So that is called as ANOVA, analysis of variance. So ANOVA or analysis of variance, this is basically used for the comparing the parameter within the different or okay, so that is called this factorial interaction effect. So option A is the correct answer. ANOVA is basically a standard statistical method which is used to interaction effect in the factorial experiment. In factorial designing, ANOVA is the individual effect, each independent variable and the joint effect of dependent variable. Okay. So GPAT 2026 marathon test is going on. So you need to join this batch GDC MAHA 26. On the next test will be on 31st of January, 9 p.m. So all are requested to join the batch. I want or everyone those who are present in the class comment down in the comment section that yes, I have joined. So I will check how many of you have joined because this all things we are planning for your betterment. Because when you will practice many exams, so I am telling you my strategy. When I was preparing for GPAT, we usually practice lot of questions. Okay, when you practice lot of question and you will able to answer this question, you get a confidence. So when you sit in actual exam, you will feel that array this question so I have seen in my mock test. So this confidence we want to develop within you. Okay. So, so in this class, we have discussed the 50 important MCQ from pharmaceutical biotechnology and pharmaceutical management and some part of the biostudies. Okay. I hope this, the 1 hour time that we spend with you in this discussion will be fruitful for your upcoming exam. And I request every students to join the batch and attend the test. Okay. So that's all for today. Thank you. Thank you so much. Bye.